Hello everyone, welcome back to my channel. Today we'll be doing biological molecules. This is second part of that chapter. In the first part we did the introduction to biological molecules, then we did water and carbohydrates. In this part we'll be moving on to proteins, lipids, nucleic acid and conjugated molecules. So without further ado, let's get started. What are proteins? They are, as we saw earlier, they are the most abundant organic molecule in a cell. In eukaryotes, they are 18%, and in prokaryotes, they are 15%. Also, they constitute 50% of the dry weight of the cell, as we mentioned earlier. They can also be known as macromolecules because they have high molecular weight. Apart from that, they are also known as heteropolymers because they constitute different type of monomers, and we'll see why they have different type of monomers later on when we see the structure of the amino acids. Okay, so what happens is we have amino acid. If, as we did earlier, condensation and hydrolysis, if we remove a water molecule, they will turn into proteins. This is condensation and also known as dehydration synthesis. And if we add a water molecule, that is hydrolysis, the proteins will get converted to amino acids once again. And what are exactly amino acids? They contain essential elements like carbon, hydrogen, oxygen and nitrogen. Now we can see here the nitrogen is extra because when we did carbohydrates, we had only CH and O. Now in proteins, this nitrogen is in addition. Some key sentences that you have to memorize is these ones. Let me first do. Okay, so 170 type of amino acids have been found to occur in cells. These are some key numbers that you have to memorize for your MCQs, because direct MCQs come from these numbers, and 25 of them are constituent of proteins. The common ones we know are 20 only, and among these 20, 10 are essential and 10 are non-essential. Non-essentials are made in the body, although this seems weird to read, but it is what it is. Non-essential ones are made in the body and essential ones are not made in the body, that's why they have to be consumed. Moving on to the structure of amino acid. This part of amino acids, NH2, CH and COH, carboxyl group, this is a constant part. And R group, that is our alkyl group, that is a non-constant part. So for example, if in R group we have an H, that will be then known as glycine. And in R group, if for example we have CH3, then that will be known as alanine. Okay, and because we have NH2 in this group, that is the amino group, this is known as the N-terminal, and we have carboxylic group in this end, that's why it is known as a C-terminal. Now these can be reversed, so you cannot be like, okay, the left one is the N-terminal and the right one is the C-terminal. That is not the case. Wherever the COH is that will be the C-terminal, and wherever the amino group will be, that will be the N-terminal. Okay. Some more key sentences that I have written here, and also some information about sickle cell hemoglobin is here. So the most abundant protein in nature is rubisco. We'll see the role of rubisco when we do bioenergetics. In blood, it is hemoglobin, as we know, that is the transport of gases. In ECM, that is the extracellular matrix, we have collagen that is present in our bones and joints and ligaments, etc. And in plasma, we have albumin. Okay, moving on to sickle cell hemoglobin. In sickle cell hemoglobin, the problem is that each beta chain, the sixth amino acid in each beta chain is replaced. And how is it replaced? Glutamic acid the sixth amino acid is glutamic acid, but it is replaced by valine. That's why we have the sickle cell disease, and our RBCs become sickle shaped. Okay. Now we'll see the formation of dipeptides, and then we'll see the different structure of protein formation. And how are dipeptides formed? And what are dipeptides? Dipeptides are basically two amino acids combined together. Two amino acids combined together. And how are they combined? By condensation. So you guys have to know what the peptide linkage is. So that's why I have shown here. For example, we have this one amino acid, we have this another one. Okay, we have this another. Now these both will combine, and then we'll have this. So what happens is the OH from this carboxyl end and the H2 from this carboxyl end as I was saying, the OH from this carboxyl end and just one H. I have not drawn, let me draw two H's here. Wait. And one hydrogen from this end will combine to form a water molecule, and here we will have this whole thing is the peptide linkage, and this will be formed. Moving on to the structures of protein. First we have the primary structure. And what is the primary structure? It is the number and sequence of amino acids in a protein molecules, the most basic structure. This is usually whenever we say that our proteins are denatured, this one is the last one to break because it has strong peptide bonds. And the example is insulin. In the secondary structure, we have it is categorized by form and shape. We have alpha helix and beta pleated sheets. The most now here it is written that in secondary structure we have hydrogen bonds, but of course we know if the secondary structure has been formed, the primary structure has already been formed. So we have peptide bond as well as hydrogen bonds, but here we are saying that the thing that stabilizes the secondary structure is the hydrogen bonds. Thus, in the tertiary structures, what happens is that there's bending and folding of polypeptide chain, and usually here we can see this type of structure, globular structure we say is formed, and we'll see this later on in detail. And then in this one we have ionic hydrogen and disulfide bases. Usually we remember it is as HID. So it's like hydrogen, ionic and disulfide bases. And the example for this is myoglobin, because it has one polypeptide chain as we remember, and we'll also see this in later on in the chapters we'll study. In the quaternary structure we have two or more polypeptide chains combined together. So we have two or more polypeptide chains held together usually by every type of bond: ionic, hydrogen disulfide and hydrophobic interactions. So in quaternary we have all the four types of bond that are present. And the example for this is hemoglobin, because we know it has four polypeptide chains that we'll also see. Moving again to the primary structure, we can say here I have written some things. What some things are that is that the primary structure, this arrow does not indicate insulin, this indicates the primary structure itself. Okay, so the primary structure was first introduced by F. Sanger in 1951. That's some key point that you have to remember. And the insulin, we have to know about the amino acids of insulin. Insulin have 51 amino acids. It has one alpha chain and one beta chain. The amino acid in alpha chain is 21 and the beta chain is 30. Now you'll say there are two polypeptide chains in this one. Why is it categorized as a primary structure? That's what it is written in the book. So we just take it as it is, because when our provincial book says something, we just take it as it is. Okay. And then, for example, now if they ask, what are the peptide linkages in this one? Like, we know the amount of amino acids the insulin has. It has 51 amino acids. But what about the peptide linkages? Are they also 51? They cannot be 51. Wait. Okay, they are asking कि हमारे पास 51 amino acids हैं. यहाँ पर हमारे पास दो amino acids थे. This one, 1 and 2. And the peptide linkages were 1. Okay, 2 amino acids, 1 peptide linkage. But here we have 51 amino acids. Will we have like 50 peptide linkages? No, that will not be the case. The formula for calculating peptide linkages will be the number of amino acids minus the polypeptide chain. So the number of amino acids we have are 51. Here you can see. And the peptide linkages we have are alpha chain and beta chain. So we have two peptide linkages, polypeptide chains. So the amount of polypeptide linkages will be 51 minus 2, 49. You guys have to remember this formula. It will be very helpful, hopefully. Moving on to the secondary structure, we have not much detail about it, but we should know that in the alpha helix, every turn of the helix have 3.6 amino acids. That's something you have to remember. Also, you have to remember that the beta pleated sheets are more stable than the alpha helix. I have not written this down, but you guys can write it down, okay? Also, this will create some congestion here, so you guys can write it down. That beta pleated sheets are usually more stable than the alpha helixes. Moving on to the tertiary one, I mentioned when the polypeptides are folded and their bend, a globular shape is formed, and the globular shape is formed because usually what happens is the inner part has amino acids that have the hydrophobic R group. Hydrophobic means like water hating. We'll say hydrophobic means water hating. It is insoluble in water. Okay. And the outer part have amino acids which are water. Wait, let me. It is this thing. The outer part have amino acids which are water loving, and that is the peripheral part, and that is hydrophilic in nature. So the overall globular structure we know is water loving. It is soluble in water. Moving on to the quaternary one, we have example of hemoglobin here, and how many amino acids does hemoglobin have? It has 574 amino acids. And how many polypeptide chains? We have 4 polypeptide chains. 4 polypeptide chains in which there are 2 alpha helixes, 2 alpha chain and 2 beta chains. So the alpha chain will have 141 amino acids and the beta chain will have 146 amino acids, and then you have to divide multiply them by 2 because they are 2 each. So we'll get 574 amino acids. And what about the peptide linkages? As I said, okay, as I said, the peptide linkages is the number of amino acids minus the polypeptide chains. So how many polypeptide chains do we have? We have 4 polypeptide chains, right? 2 and 2, 4. So 574 minus 4, we have 570 peptide linkages. You guys have to remember this. Now we have two types of proteins. One is fibrous protein and the other is globular. We saw the globular one here. We also said that it is soluble in water. It is written right here. The shape is spherical. We can see that. Here, the shape of fibrous protein is fibrils. Basically, fibrils means strands like this. If you have strands like this, these are represented by beta pleated sheets. And what are beta pleated sheets? Wait, what is happening to my... Okay. So we have beta pleated sheets. And what protein structure are beta pleated sheets? It is secondary. So fibrous proteins are secondary in nature. Secondary structure have a secondary structure of protein. They are insoluble. They are non-crystalline, inelastic in nature, and they play a structure role. What is structure role? As we can see, silk fibers, keratin, myosin tails, these are all structure roles. Because, for example, we have keratin in hair, and that is just a structure, no functional role. And also, fibrous proteins are more stable. We have to remember that. Well, the globular proteins, on the other hand, have a tertiary or quaternary structure. We can say the tertiary, and then one more, we can have a quaternary structure as well. They are soluble in nature and can be crystallized. They are elastic in nature and play a functional role. However, they are less stable than the fibrous proteins. For example, enzymes, antibodies, and hormones, they play a functional role, as we know. For example, antibodies in immunity, that's a functional role, right? Also, hormones have a functional role as well. There are different type of hormones that we'll see in control and coordination and nervous coordination. Now we are done with proteins. Moving on to lipids. What are lipids? They are a heterogeneous mixture of organic molecules which are hydrophobic. Hydrophobic. Wait, let me do it. It's like hydrophobic and lipophilic. Hydro basically means water, and phobic we say meant by insoluble. So they are insoluble in water. They are also soluble in organic solvents like lipids, etc. So they are lipophilic. Also, something guys need to know is that they store double the amount of energy than carbohydrates, because they have more CH bond. The ratio of CH bonds is greater, and the oxygen composition is very less as compared to carbohydrates. Also, you guys need to remember that, okay, as I was saying, first our body, with the usage in our body is first carbs, then our body uses lipids, and then the last proteins are used. So if carbs run out, we move on to lipids, and then if lipids are used, we are moving on to proteins. Okay. Now we come to the classification of lipids. First of all, we have simple lipids. What are simple lipids? Simple lipids are basically esters. I don't know what is some. So simple lipids are esters of fatty acids with various alcohols. Water esters, we all know they have this function with COO. What are fatty acids? Basically long chain carboxylic acids, can be saturated or unsaturated. Long chain carboxylic acids with various alcohols have a OH function of this will combine with the carboxylic acid or fatty acid, and we'll get a simple lipid. The example of simple lipids include acyl-based salts and waxes. So what are acyl-based salts? First of all, we'll see that, and then we'll see the waxes. Acyl salts, you need to remember that they are the most abundant form of lipids in the human body, and they are esters of fatty acids and glycerol. Here we see that simple lipids are esters of fatty acids with various alcohols. In this, the alcohol is identified, which is glycerol. Glycerol is a triol, and the structure for glycerol is this. Okay, so we have a 3-carbon carboxylic acid with three OH groups, and that means it is a triene. The formula for this is C3H8O3. Okay. So if you have like one fatty acid, plus for example, if we add one fatty acid to this OH, the C we'll have a COO here. We'll have a CO ester bond here with just one fatty acid. Then we'll get a monoacyl glycerol. So one fatty acid with a glycerol, we'll get monoacyl glycerol, and then two fatty acid plus glycerol, we get a diacyl glycerol. And if we use all these three OH groups, then we'll have a triacyl glycerol. And what is triacyl glycerol? Triacyl glycerol is also known as neutral lipids. Moving on to waxes. Waxes are highly hydrophobic and have odd number of carbon atoms, odd number of carbon, and also long number of carbon, like very lengthy chain, varying from C25 to 35. Odd number of carbon atoms, highly hydrophobic, not at all soluble in water. So we have two types. There are like natural waxes, and then we have synthetic waxes. Natural waxes include beeswax, which comes from honeycomb, and we have cutin, which comes from leaf surfaces of plants. This highlighter is so struggling. Okay. And also we have synthetic waxes. They are derived from petroleum or polyethylene, and the examples include paraffin wax. That's all you need to know about simple lipids and its examples. Now, heading towards complex lipids. Complex lipids are esters of fatty acids and alcohols. That is the definition of simple lipids now, right? So we have esters of fatty acids, alcohols, and then we have an additional group. What can be that additional group? We'll see. Examples include phospholipids, which are derivatives of phosphatidic acid. What is phosphatidic acid? It is basically the simplest type of phospholipids. You don't need to remember the structure too much about it, and don't need to know too much about it. Okay. So what happens is, how is phospholipids formed? Basically, we'll have phosphatidic acid plus nitrogenous base. This will form the phospholipid. And there are different types of substances that can be added to phosphatidic acid. For example, choline, ethanol, amine, inositol, and serine. And we have to remember, phosphatidic acid is added to choline, then we'll get this another compound which is known as phosphatidylcholine, and then the common name for this is lecithin, and it is the most common phospholipid. We saw that phosphatidic acid is the simplest, and this one is the most common. You guys need to remember these names. That's all about the complex lipids. Moving on to derived lipids. What are derived lipids, and where are they derived from? They're derived from simple and complex lipids, as simple as okay. Derived lipids include terpenes. What are terpenes? They are made up of simple repeating units, can be categorized as polymers as well. Terpenes are the ones which we can say that lipids are polymers because of terpenes. Okay, they are made up of simple repeating units, and what are those repeating units? They are known as isoprenoid units, and they are usually 5 carbon compounds. These isoprenoid units. So if we have 2 isoprene units, we'll have a monoterpene. Like for example, we saw in peptide linkages, if we have 2 amino acids, then we'll have a then we'll have a monopeptide, and then we have 4 amino acids, then we'll have a dipeptide, and etc. like that. So if we have 2 isoprene units, we have a monoterpene. If we have four, we have a diterpene, and then if we have multiple isoterpenes, then we will make a then we will make polyterpene. Then also for complex derived lipids, we have steroids. What are steroids? We have a structure here as well. Steroids are 17 carbon compounds. You need to remember these numbers. 17 carbon compounds and 4 attachments, as you can see here. 1, 2, 3, 4. And 3 rings are 6 carbon. 3 rings are 6 carbon, as you can see these three. 1, 2, 3. And then one ring is 5 carbon. And that is this one, the fourth one, is the 5 carbon one. And you can see 17 carbons here. What is cholesterol is a wait, let me do it first and then cholesterol is a precursor of a large number of equally important lipids. What does that even mean? It means cholesterol is a precursor. Basically means like a father. So due to cholesterol, these bile acids, testosterone and estrogen and progesterone are made. That's why cholesterol is basically the father. All these are made from cholesterol. And cholesterol is also classified as a steroid. Moving on to the last part of lipids, it is prostaglandins. And what are prostaglandins? Prostaglandins are basically, we have all heard about this term. It acts as a local hormone. It is derived from arachidonic acid, arachidonic acid, and some of the functions of prostaglandins is that it regulates BP, induces liver inflammation, increases the sensation of pain, and also results in aggregation of platelets. So basically, if we take anti-fever medicine, that is antipyretic medicines, they depend on how well they inhibit the function of prostaglandins and reduce the fever. Okay. Moving on to nucleic acids. First of all, in nucleic acids, we have nucleotides, the basic uh, structure of nucleic acids are made up of nucleotides. We have our very famous ATP, which is known as adenosine triphosphate. We have NAD. We have all heard about these terms, right? We have nicotinamide adenine dinucleotide, and then we just add phosphate to that. We get NADP. We have FAD. They're all derivatives. Also, we have to know that they're all derivatives of vitamins. We did this in enzyme too, that all the coenzymes are derivatives of vitamins. Right. Now we have DNA and RNA. Some important information that we need to know about DNA is this. What is DNA deoxyribonucleic acid? In 1951, Erwin Sherlock provided data about the ratio of different bases in DNA. What are the different bases in DNA we can see here? We have nucleotide. What am I doing? Okay. Here. We have DNA and RNA here. The nucleotides that make up DNA is deoxyribonucleotide, and for RNA it's ribonucleotide. The bendo sugar deoxyribose, and here it is ribose. So here we're talking about the nitrogenous bases AGCD, which is adenine, guanine, cytosine, and thymine. So here Aaron Sherlock, what he provided data about the ratio of different bases in DNA. Double helix, like DNA is a double helix like this, and the strands are anti-parallel. You know about that. So we have purines and we have pyrimidines. What are pyrimidines? The easy way to remember this is known as cut. This is cytosine. I forgot the uracil and thymine. We won't include uracil in the DNA, we'll include it in the RNA. For adenine and glycosine, this is not glycosine, it is It's guanine. Why have I written glycosyl? Okay, it's guanine. I don't have that marker here. Guanine. Okay. So we have adenine and guanine in the purines. The purines are bigger bases. They have two rings. Some more info that you guys need to remember. These are smaller ones and have only one ring. One ring. So the way I remembered these one was like the pyrimidines are like longer, but this is the opposite of like the pyrimidines are actually smaller bases, but actually the letters in this pyrimidines are far more than in purines. That's how I remember it. It like you guys can remember it however you guys want. The abbreviation, the mnemonic for this one was as pure as gold. So the as gold here was adenine and guanine, and pure here was for purines. That's how I remember it. Okay. Some more key sentences to remember. I don't know why these people ask these type of things, but they do. That Haemophilus influenza was the first microbe to have its genome sequenced completely. You guys need to remember. Also, we have the bases in RNA. RNA do not have thymine. It has uracil instead of thymine, but the bases are 4 in each. DNA is double-stranded. RNA is single-stranded. It is also synthesized by DNA. RNA, here I have written this thing. In the process of transcription. We'll do that later on as well. The location of DNA is or has chromosomes, nuclei, mitochondria, and chloroplast. The same is for RNA. It has nucleolus. Mitochondria and chloroplast is the same. Ribosomes and cytosol are the extroverts. The amount of DNA is constant in cell of the same species. You need to remember it is written same species. Different species may have different type of different amount of DNA, but it is constant in each cell of the same species. However, RNA is variable from cell to cell. The role of DNA is hereditary, carries genetic information as genes, and in RNA it is protein synthesis, for example, transcription and translation. So here we have a table of 3 different type of RNAs. We have messenger RNA, we have transfer RNA, and we have ribosomal RNA. The function of mRNA is to take message from DNA to ribosomes. DNA is present in the nucleus and then it transfers it to ribosomes for translation process. It is single strand of variable length. Variable, how is it dependent on the size of the gene? Okay, it depends, the length depends on the size of the gene. It is variable. Also, for example, if we have one amino acid, is coded by 3 nucleotides. Okay, so if we have an mRNA of 100 amino acids, how many nucleotides will we have? 300. Okay. Then you need to remember these percentages. mRNA is 3 to 4%. That is like in the RNA which is present in the least amount is mRNA. Then it is tRNA because we have 10 to 20%. Then it is ribosomal RNA which is 80%. What is the function of tRNA? It transfers amino acid to ribosomes during the process of translation. Its length is very important. I'll highlight it in different color. Its length is of 75 to 90 nucleotides long. This is also an MCQ. RNA is used in the formation of ribosomes, and it is a double helix with constant length. Also remember the percentage, it is 18% in this cell. Okay. Moving on to our last topic, which is conjugated molecules. What are conjugated molecules? They are made up of different type of molecules combined together. to form one molecule. So for example, if we have carbohydrates and proteins, right? Where carbohydrates and proteins, they'll combine to form glycoproteins. And what are the roles in which glycoproteins are used? They are used in cellular secretions. This one is important. Cellular secretions. They are integral part of membranes. They are used as enzymes. They are used as hormones transport and as transport and structural protein, also as receptors on the cell surface membrane. And then we have carbohydrates and lipids. What are carbohydrates and lipids called? Combined together, glycolipids. They're also integral part of membranes and brain, white matter of brain, and myelin sheet of nerve, a fiber. Lipids and proteins combined form lipoproteins, part of milk, blood, egg yolk, membrane, and cell nucleus. Everything important in this story. What are nucleic acid and histones called? They are called nucleostones. If I can. I'm not able to extend this thing. Okay. So let me try again. No, let's not do that. Okay. We have nucleostones. They are found in the ribosomes, chromosome, involved in regulation of gene expression. Because as you can see, histones are part of DNA, and then we have nucleic acids. So they are involved in regulation of gene expression. That brings us to an end of biological molecules. I hope you guys have understood everything. This is all that I managed to incorporate in this lecture. I covered as many things as possible so that you guys don't have to read the books again and again, and you can just open your notes. I hope you guys are making notes because I think that's the best way to do to practice for MDCAT. Make notes, attempt MCQs afterwards, and then do message me if you guys are having any problems with MCQs, and I can make a video about that too. Now, I will be making another videos about different chapters. For now, that's it. Allah Hafiz.